learn more about RNA sequencing from Professor Angela Brooks and graduate student Cameron Soulette.
Thank you so much, George Kraw, for helping to support this lecture series and the technology and science going on at UC Santa Cruz. So thank you so much. And thank you all for coming. It's really great to see new people and old friends as well. So I'm Angela Brooks. I'm a junior faculty at UC Santa Cruz. I've been here for about 3.5 years. And I'm going to talk about the work going on in my lab kind of broadly, and then I'll hand it over to my talented graduate student, Cameron Soulette, who will talk more about his project in more detail. So my lab is interested in studying cancer, and we study lots of different cancer types, but one particular cancer type we focus on is lung cancer. And one of the main reasons is that it is the leading cause of cancer deaths in the US. And so we're interested in understanding the biology of lung cancer, and also by understanding the biology of lung cancer, thinking of ways to possibly treat more individuals with this disease. So before I actually got into the cancer field as a postdoc, but before my postdoc, I actually had my first introduction to lung cancer and lung cancer treatment through a friend of mine named Maki Inada. So this is a picture of her and her family. And when I was a graduate student at UC Berkeley, Maki was a postdoc in the same lab. And after she left the lab, she was looking at positions in independent scientist position. And she was in her mid-30s, and she actually was diagnosed with lung cancer. And she never smoked, and so that was also at the time really surprising. And so her doctor realized that since she was of East Asian descent and also a never smoker, that commonly those individuals tend to have mutations in a gene called EGFR. So through DNA sequencing, they sequenced the version of EGFR in Maki's lung tumor, and they identified a mutation that was known to cause a change in this EGFR gene. And fortunately, there was a known inhibitor that could specifically treat patients that had this type of mutation. And so how do we get to that point? How do we get from finding a gene mutation and understanding what, or deciding on what kind of treatment to provide for those patients? So often, and now for actually a lot of hospitals, when an individual comes in with lung cancer, they can have part of their DNA or sometimes even their whole genome sequenced. And then if they take the tumor DNA and sequence it, and then compare that DNA to the normal DNA, say from a blood sample, and they can look for positions in the genome that are different in the tumor versus normal, and those are potentially mutations that are contributing to their cancer. And then when you sequence many patients that have the same type of cancer, say for lung cancer, and an example in a study I was involved in where we sequenced about 500 lung adenocarcinomas and then looked at what genes were mutated, you can see that about 30% of these lung cancers had mutations in a gene called KRAS, and about 11% have mutations in this gene EGFR, which was the same gene I mentioned that Maki had. And so when we sequence many patients, we can see what are the genes that are commonly mutated across many different individuals, and those would be the most likely genes to try to identify ways to treat because they're commonly found in many individuals. So if we can find a matching treatment for those patients, you can impact a larger set of patients. So before I go into how we think about treating these types of cancers, I just want to go through kind of the central dogma of molecular biology and what we do with that information of a gene mutation. So if some of you remember, the central dogma of molecular biology is DNA goes to mRNA that gets then translated into protein, and the proteins usually are the things that performs the functions in your cells. So here I'm diagramming that a piece of DNA, which is the genotype or the sequence that an individual has. That DNA is transcribed into mRNA. It's a single-stranded RNA molecule. And then that RNA is then translated by ribosomes into protein. And then that protein then has some biological function. So what happens with cancer, often what happens, is if there's a cancer-causing mutation, that mutation will change the sequence or the instructions at the DNA level. And then those instructions will then get copied into the mRNA. And that can lead to a different code for what amino acids should be at that position. And so you end up with a different protein sequence. And then that can lead to a different biological function of that protein, which can possibly lead to cancer. And so in the case of, say, the EGFR gene, this is one example that there are mutations that are known to affect the protein of the mutation then leads to a difference in the protein sequence of EGFR. And these EGFR inhibitors are often these inhibitors that target these cancer mutations. Ideally, we select inhibitors. These can be small molecule drugs. Sometimes they're antibodies that target those specific proteins. And ideally, we want to find inhibitors that are specific to the mutated form of the gene, but do not affect cells or the version of the protein that's in normal cells, because you wouldn't want to inhibit or hurt your normal cells. And so often when we think about targeted therapy, we're trying to identify these kind of cases. Okay, so again, we sequence lots of different cancer genomes so we can identify the mutations in commonly mutated genes, and that can maybe help to lead to think about what are potential targeted therapies for those individuals. One challenge, though, is that in the case of lung cancer, in the case of many other cancer types, most of the mutations that exist are actually quite rare. So less than 10% of patients have the same exact mutation. And so the goal of my lab overall is to really understand out of the sea of all these millions of cancer mutations that we're observing, which ones can we use for treatment. And first, we want to understand which of these mutations are actually contributing to cancer. So a lot of these mutations can happen randomly and maybe hit a region of the genome that actually doesn't function at all or doesn't change the cell. And so it's hard to actually pick out which of those mutations are actually functioning and which ones might actually be functioning. And so some of the questions we ask are which of these mutations we're seeing in patients contribute to cancer, and out of those mutations, which ones can actually be informative for precision medicine. So the approach that my lab takes is to look at what's called the transcriptome. And so to make sense of these cancer mutations, we study what we view of as the output of the genomes, which is the transcriptome. So what do I mean by that? So most people, when I talk about what I do, have heard about DNA and genomes, potentially. So genomes is the collection of all the DNA in our body. But what actually is interesting to think about, and this was something when I first learned about was kind of mind-blowing to me, but every cell in your body, every tissue, has exact same DNA, the same instructions of how to make a liver cell, a muscle cell, brain cell, and so forth. And so how do you have that same DNA, but then have such different cell types? And that's because of the RNA. So in each of these different cell types, and here I'm just talking about normal tissue, they have different outputs of the genome. So the different RNAs are being expressed in these different cell types, and that's what helps define what the function of the cell is. And so in the case of cancer, what we think happens is that these cancer-causing mutations are most likely going to change the transcriptome, or what the output is of the genome. So for example, if you have a somatic mutation, which is a mutation that arises in it's a mutation that you did not inherit from your parents, but something that arose in one of your cells in a specific tissue. So if you have a somatic mutation, say in a liver cell, then that might change. If it's a cancer-causing mutation, it's likely to change the output of that cell. And so you might see different transcripts that are expressed in that cell. And in some cases, these specific transcripts might actually contribute to changing that normal liver cell into cancerous cells. So these RNAs can either directly function in affecting a cell to become, come from normal to cancer, or these RNAs can also be used as what's called biomarkers. And so these can be used to correlate patient prognosis, or correlate maybe effects of different therapies. So these RNAs can be used in many different ways. Okay, so going back, so again, my lab is interested in looking at the mutations and understanding them in terms of how they're changing the output of the genome, which is the transcriptome. And another reason why we're interested in the transcriptome is because after sequencing many different cancers, there are a set of genes, they're called splicing factors, that are, in the case of lung cancer, rarely mutated, but in this sort of pool of genes that are rare. And so these splicing factor genes are already known to affect the transcriptome, and so we were interested in investigating these type of genes first. So what is a splicing factor gene? So before I go into splicing factor genes, I do want to mention the prevalence of these type of mutations. So here's a chart from a review a couple years ago of the prevalence of these splicing factor gene mutations across many different cancer types. So they're actually most frequent in hematopoietic malignancies, which are blood cancers. And my lab actually collaborates with a group that studies chronic lymphocytic leukemia. And so that's one cancer type we also study. But today we're going to focus on the lung cancer type, which has, again, very rare but frequent mutations in these splicing factors. So they occur pretty frequently. And so what is splicing factors? So these are genes that control the process of this DNA to RNA to protein. And the step that they actually affect is actually after transcription in this step called RNA splicing. So after DNA gets transcribed into what's called a pre-messenger RNA, it then gets spliced. So what is RNA splicing? And moreover, there's a process called alternative splicing. So about 95% of our human genes go through this process. So it's very, very frequent that we have this type of gene processing. So here I'm diagramming a gene, so kind of the DNA portion, a segment of the genome. And that gene at the DNA level is initially transcribed into a pre-messenger RNA. And the boxes here, I'm cartooning those segments as what's called exons. And the lines in between are called introns. And so alternative splicing allows one single gene message to then be alternatively processed into many different products. So you can have just every intron, each black line spliced out and joined together to produce this one long, say, transcript that might be specific to the lung tissue. You can also decide to skip over exons, as shown here in the skeletal muscle example, where you don't have that blue exon anymore. Or you can decide to skip multiple exons and have a completely different transcript. And so, again, about 95% of our human genes go through this process. And why that's potentially interesting for diversity of gene function is that you can have different gene functions from the same initial message. So it allows for kind of greater control of gene regulation and also potentially arising new functions of genes. Okay, so how are these decisions made? So how does a cell know where to cut and paste together? And so it's actually these splicing factor genes that help control this process. So here I'm just showing, again, 2 exons as boxes and then an intervening intron as just a line. And the 2 factors that we study in the lab, one is called SF3B1, and then the other one is called U2AF1. And these factors actually associate directly to the pre-messenger RNA and help direct the cell to know exactly where to cut and paste. And it's these factors that are commonly and frequently mutated in cancer. And so this is such a fundamental process to how cells should work normally. And so it was very curious as to why these mutations were occurring so frequently in cancer. Yes? Where do these factors come from? Yeah, so exactly. So these factors, the genes for those factors are encoded in our genome, in some other part of the genome, and then these get mutated. So some point mutation that causes then a change in their protein sequence, and then so when they try to go and do their normal function in the cell, they're going to affect other genes. Mm-hmm. And so this is a big question in the field, even still today, is really understanding why are these splicing factors mutated so frequently. And the view that we take is we think that because these splicing factors are known to control how this splicing process functions and where to cut and paste in the pre-mRNA, we're interested in looking at cases where this gene is mutated and then what the downstream effects of the RNA are. So we're interested in looking for maybe you see exons that are skipped when they shouldn't be in the presence of these mutations. Or maybe you see introns that are completely not spliced because you're, again, mutating the splicing factor, it can't find where to splice out the intron. Or we might expect maybe other kinds of RNA changes that might occur. And so we're interested in understanding the collection and what are all the transcriptome changes that are occurring because of these gene mutations. And if we understand that, we might be able to identify those genes that are affected by this process and potentially some gene pathways. And so a lot of known cancer mutations kind of hit the same type of, say, growth or proliferation pathways or cell cycle pathways. And so we're trying to understand which of these cancer pathways might be hit by through changing the RNA. Okay, so our approach to try to identify the RNAs that are changing because of these splicing factor mutations is to actually go straight to patient RNA data. Through collaborations or publicly available data sets, we can take donor samples that have no mutations in these splicing factors, and then take another set of donors that actually do have a mutation in these splicing factors. And then we can take the actual sequencing of the RNA. So some of you might have heard of DNA sequencing, where you can actually sequence the RNA as well. So we can take that RNA sequencing from these 2 sets of donors and then look for differences in splicing. So what introns are being spliced out in that's different in the donors that have these splicing factor mutations. So the reason why we can do this is often, and not as often as DNA sequencing, but there are, I think, more and more clinicians that are also not only taking DNA sequencing from these tumor samples, but they can also get the RNA sequence. So from, say, the section of the tumor, we can then grab those cells and extract the RNA from those cells and sequence the RNA to understand what RNAs are expressed. And in some cases, you can even get adjacent normal cells. So these are cells that are kind of nearby the tumor. They're still lung cells, but they shouldn't be cancerous. And we can sequence also those RNAs. And then by comparing the normal RNA to the tumor RNA, we can identify potential transcripts like that shown here in red that is specific to the tumor and not observed in the normal. And those will be candidates for transcripts that are altered in the tumor and not in the normal, and maybe point to a transcript that might be contributing to the tumor formation. Okay, so again, since we have this RNA data collected from a large group of donors for different cancer types, we can look at, again, what are the differences in splicing because we have that RNA sequencing. So most of my actually postdoc work was to look at the splicing changes that were caused by these splicing factor mutations in primary samples. But there's a limitation to this type of sequencing that most people or most researchers use right now. So typical RNA sequencing will take this, the RNA, and we usually look at a population called poly A plus or polyadenylated RNA. Those tend to be the protein coding RNAs. And so we take that population of RNA, and then we actually have to make a copy of it into DNA. And that's what's this cDNA is complementary DNA. So it's a DNA sequence that's a copy of the RNA. And then we have to cut it up into short pieces. And that's basically the one of the technologies that's used for sequencing. You need to have the segments kind of short because you can only read a little piece of the DNA. And so one challenge with that, though, is because we're cutting up the sequence into little pieces, when what we get out is just a little chunk of what the sequence was in that RNA. And so matching it back, like let's say if we sequence this little chunk here, it's really difficult to know exactly which RNA was present because that little gray segment looks really similar to this RNA transcript, but it also looks very similar to this one. But they have different patterns of splicing, like shown here. The intron here is kind of bigger than the intron here. So these are actually different transcripts that are expressed. So again, the short read sequencing or short read cDNA sequencing makes it really difficult to know exactly what RNAs were actually present. And so, again, I mentioned in my postdoc work, this is what I primarily focused on, this short read sequencing. And I knew when I was going to start my lab, I was really excited about upcoming technologies that could actually sequence longer segments of DNA. And so what a lot of my initial projects in the lab that we launched was to utilize long read sequencing technology, where we can take the poly-A RNA, but then make a DNA copy, and then but not have to cut it up into smaller pieces, so we can actually see the entire full-length copy of that RNA. So we have a better sense of exactly which RNAs were expressed initially. And so I was particularly excited about coming to Santa Cruz because there was this new sequencing technology that was actually invented here by David Deemer and other colleagues called Nanopore sequencing. So if people have heard, I think Mark Akison talked about this type of sequencing, maybe it was last year. And so I actually brought the device so you can kind of get a scale of how small this is. So this is the same thing, maybe kind of, that's probably an older version of it. But it's very small. It looks like the size of a USB drive. And you connect it directly to a laptop. And you can sequence, so you have to order flow cells that go into the device, and then you load your samples in, and you can sequence very long reads with it. And so feel free after the talk if you guys want to look at that a little bit more. So this allows us, this type of technology allows us to sequence the full transcripts that are present. And so some initial projects in our lab, we wanted to understand the effects of the mutations of these two splicing factors, one called SF3B1 and another called U2AF1, using this long-read technology so that we could see what are the actual full-length transcripts that are being altered as a consequence of these mutations. So the first project was led by Cameron Soulette, looking at U2AF1, and he's here and he'll talk more about his project. And the other splicing factor our lab looks at is called SF3B1, and that project is led by Allison Tang, who's another graduate student in the lab. And the work from that study is actually available on what's called bioRxiv. I don't know if others have heard about this. It's kind of a preprint server, so people can look at the results of our initial studies. So basically, we submit that to the preprint server, and then we submit for peer review at a journal. So right now, that paper is under review. But I wanted to talk. I wanted to bring Cameron to talk about all of our unpublished data for this U2AF1 gene and what we've been doing with Nanopore to investigate those mutations. So I'd like to bring up Cameron Soulette. So he's a PhD student in molecular cell and developmental biology, and he's here to talk about his project.
Awesome. Thank you, Angela. I think we got too close together. Let me just jump right here. All right. Thank you, everyone, for being here tonight. What I'll be talking about is just a small part of my thesis project, which is using new sequencing technologies that Angela just told you about to investigate mutations that occur in the splicing factor called U2AF1. And we're interested in looking into the splicing factor and the mutations that are associated with it because it leads to transcriptome-wide splicing alterations. And just to start out, I want to show you this quick animation of this first step of gene expression, transcription, and pre-mRNA splicing, because I think this really nicely illustrates what's actually going on in the cell and how these proteins interact with DNA and with the pre-mRNA. So here we're looking at the first step of gene expression, transcription. The strand that's being synthesized is the pre-mRNA. The two different color strands over here that you're seeing are exons and introns, and these 3D blobs kind of floating around are the splicing factor proteins. And as you can see, the splicing factors are binding on to the intronic sequence at the boundaries, bringing them together and removing them from the pre-mRNA transcript to make the fully mature mRNA transcript. And so, again, just to go over a little of the vocab, so let me see if I can make this thing work. Oh, cool, sorry about that. It's too fancy for me. All right, so these orange parts are the exons here, green parts are the introns, and as I've already mentioned, these 3D blobs here are the splicing factors, and my project looks specifically into this splicing factor here called U2AF1, which is important in binding the 3' boundary, one of the boundaries of this intronic sequence. And so we're interested in studying this splicing factor for reasons Angela's already mentioned. It's recurrently mutated in various cancer types. What I'm showing you here are the different types of mutations that are occurring across different cancer types. And at the bottom, I have kind of an idea of what the protein sequence looks like for U2AF1. If you were to take that 3D structure and just flatten it out into a string, this is basically just the protein sequence of U2AF1. And so, as you can see, there's this mutation that's kind of jetting out from the rest, which is this S34F mutation. This is the mutation that we look at, and we're currently studying this mutation and how it impacts U2AF1 in the context of lung adenocarcinoma. And so the big question that we're trying to answer for all the splicing factors, but also for this U2AF1 splicing factor, is how does this mutation actually promote cancer? Why is it being selected for in tumor cells, and how is it advantageous? So to answer that question, we have to look at what the function of normal U2AF1 is. So in cells with normal copies of U2AF1, it will bind onto all of these intronic sequences for every single mRNA in the cell, so that it can effectively splice them out and include all of the necessary exons for that transcript, so it can go on to be translated into a functional protein. In contrast, when we have mutant copies of U2AF1, sometimes these intronic sequences don't get recognized. And when these intronic sequences don't get recognized, there are certain exons that don't get included in the final mRNA transcript. And this can occur in a variety of different combinations. And so you get these alternatively spliced products that are only seen in the presence of this mutation. And so a question that remains unanswered that we're interested in following up on is, what are the function of these mRNAs that we only see in the presence of this mutation? Could they possibly be the providing some advantageous characteristic to tumor environments? And so our working hypothesis right now is that these mRNA isoforms associated with this mutant U2AF1 are somehow promoting tumorigenesis. And so how do we answer this question? Well, we have to do some sequencing, of course. Here's some sequencing data. And I know it's a little late, so I'm going to try to wake you guys up with the short quiz. Everyone's going to put on their scientist hat tonight. And what I want you to do, here are the mRNA isoforms. Here is real data from a real sequencing experiment. We have short reads here and long reads here. And I want you to tell me what's easier to basically assign to an isoform. Is it these short reads or is it these longer reads? Right. So for reasons Angela's already pointed out, these short fragments are really hard to assign to any individual read. And when your goal is to characterize which isoform is being expressed and to measure its actual expression, it's hard to divvy up these short fragments to the number of isoforms that could be derived from some gene. But so the question then arises, like, why don't we always use long-read sequencing? Well, there's a number of factors why we don't just use long-read sequencing. There's many differences between the two technologies. And mainly, the differences lie in these three criteria, which are size, throughput, and error rate. So size just describes how big of a molecule you can sequence using these technologies. So for short-read sequencing, a pretty standard protocol will sequence fragments up to 150 base pairs. For long-read sequencing, depending on the strategy, you can sequence anywhere from 300 base pairs up to 1 million base pairs. For throughput, this just describes how much data you can get out of a sequencing experiment. How many reads or molecules can you actually sequence? And this number is pretty different between the two technologies, as you can see. For short read, you get around 350 million reads or 350 molecules sequenced, whereas for long read sequencing, you get anywhere in the range of 1 to 10 million, depending on the technology. So you get a lot more reads for short. read sequencing. And error rate, probably the most important, depending on what type of analysis you're doing. For short read sequencing, I think this number is actually inaccurate. I think it's smaller than 0.001%, but you can kind of see the disparity between these two numbers, which is that in long read sequencing, you get an error introduced into your sequencing one time out of every bases that are called. So from these criteria, it might look like that you want to use short read sequencing because you get a ton of data and it's really accurate. But for the type of analysis that we're doing, which is characterizing these long mRNAs, the size really kind of trumps everything. So for these reasons, we've chosen to use long read sequencing for this analysis, mainly for its advantage of sequencing long molecules, but also because we have a great resource here at UC Santa Cruz. David Deemer and Mark Akison are kind of the godfathers of this technology. And they've actually built a really strong community of Nanopore users at UC Santa Cruz, which has been really great to actually get this project up and running. So I know Mark was already here and explained probably how this worked, but just to give you a refresher, the way nanopore sequencing works is you have this nano-sized hole that's embedded within a membrane. And to sequence your molecule of interest, you just basically pass this molecule through this nano-sized hole using ionic current. And as your molecule is being passed through this hole, changes in ionic current are being measured with electrodes, and those changes in current are being translated into the different nucleotides that comprise that molecule. And so going back to the main purpose of my experiment, again, I'm studying the splicing factor that creates a whole bunch of novel mRNA isoforms that we want to identify and quantify, and eventually test what their functions are. So to do this, we're using a lung cancer model cell line, these human bronchial epithelial cells. We have two cell isolates. We have a wild type copy, which has the normal versions of this U2AF1 gene. And then we have a mutant isolate, which has one copy of a mutant U2AF1. And so what we do is we just grow up these cells, we harvest their RNA, sequence them on the MinION, and then analyze the data using an algorithm that we've developed called FLAIR. And so first, I just want to run through the numbers of how our sequencing experiment actually went. So these are just numbers from our 6 sequencing experiments. We sequenced wild type 3 times and mutant 3 times. And across all 6 experiments, we got somewhere around 1 million reads per sequencing run. We did these sequencing runs last year, and so these numbers are kind of low in comparison to what people are getting now. The data or the technology is rapidly evolving, so a lot of labs and collaborators that we have are getting read counts that are up to 10 million and over. And so, but nevertheless, 1 million reads per condition was more than enough to actually go ahead, characterize isoforms, and do the quantification. So we were very happy with these numbers. And the next question that we had was, we have 1 million reads, but are they long? Do they actually look like human mRNAs? So what we looked at was the read length distribution and compared it to what the average read or what the average mRNA length is for human. And so as you can see, we have this nice distribution that has an average somewhere around 1 kb or 1,000 bases, which is pretty close to what the average human mRNA length actually is. So that was nice to see. And so now that we have enough data and we know that the reads are long and they look like actual mRNAs, we take this data and run it through this algorithm that we've developed. For short read sequencing, it's been around for over a decade, and there's a huge community that has built a lot of tools to analyze short read data, but long read data is relatively new. So there's not a lot of tools out there, and the few that are out there are very hard to run. So we developed our own algorithm that can basically take raw nanopore data and convert it into assembled isoforms that we can quantify against. And this project was spearheaded by a very talented graduate student, Allison Tang, And if you're interested in how this algorithm works, we have a paper on bioRxiv. So we wanted to identify changes in gene expression in our long reads between mutant and wild type. So there's really two outcomes to this type of analysis, and it's a very straightforward analysis. After we have our isoforms, we just count how many reads map to each isoform. So one example is that there's no change between mutant and wild type, where for this particular isoform, there's a similar number of read counts for that isoform. The alternative is that there is a significant change where in wild type there might be a low count or a low number of reads mapping to this isoform versus mutant where we have high expression of this isoform. And so we went ahead and did this test and tested over 59,000 isoforms. We found that 360 had significant changes in expression. And we were just curious, because we're using this long read data, how many of the isoforms that we identified are actually novel, are actually not found in any other annotation set, or they weren't even found in previous short read experiments. So from our 306 isoforms that had significant gene expression, or isoform expression, we found that nearly 2/3 of them were actually novel. So we would have missed these if we just had used regular annotations or short read data, which is really, really interesting. And so I'm going to just highlight one gene that came out of this analysis, which is this PTMA gene. Right now it's classified as a biomarker, which just means that it's associated with some characteristic of a tumor and like perhaps the aggressiveness of that tumor, but the biological role of this gene in that trait isn't well understood. But it is a putative oncogene. There are some functions that are known about PTMA, which is that it's associated with poor prognosis in different cancers. It's implicated as an anti-apoptotic factor, which means that it kind of goes against cell death. It helps the cells stay alive. And it's also associated with chemoradiation resistance. And so this is an interesting gene that we want to follow up on and perhaps take some of these isoforms and test their function individually. To the right, I just wanted to highlight again the novelty of using not FLAIR, but nanopore sequencing, which is that we were able to identify a whole bunch of isoforms that had significant changes in expression. As you can see here, so these are the isoforms that came out of our algorithm, and on the top are the annotations that have been previously published. And so as you can see that there's a lot more isoforms that we're discovering using our algorithm. So as I mentioned, our next steps are taking these 306 isoforms, well, not all 306, but some of them, and then doing some type of integration into a wild type background, and then measure out any changes in things like radiation resistance or anti-apoptotic potential. So with that, there's a lot of people who helped get this work off the ground. My PI, Angela Brooks, obviously, Allison Tang, who put a great deal of effort into building this FLAIR algorithm, the Akison lab for being at UC Santa Cruz and helping us work out the nanopore and give us good results, and you guys for listening. Thank you.
So I just have a last little bit, and then we can go to questions for both of us. So everything we've talked about so far is what we've been really excited about with doing cDNA sequencing with the nanopore. But actually, one of the reasons, so there are other competing long-read technologies, so you can do the cDNA sequencing with other platforms. But the main reason why I was really excited about Nanopore was because of its potential for actually sequencing RNA molecules directly. So I'm going to talk about some preliminary work of a collaboration of where we've been using this technology to actually sequence RNA directly. Okay, so why would we want to sequence RNA directly, and what are we missing still from the cDNA sequencing? So I'll go back to this diagram I showed earlier, which is usually with Nanopore cDNA sequencing, all the work that Cameron described, we get a lot of information because we're getting the full transcript. But unfortunately, we have to copy the RNA into DNA. And so, but actually, RNA can be modified. So there's an expanded sort of information that exists on RNA, and these modifications are known to affect how the gene gets regulated or processed. Maybe this M6A, for example, this M6A modification, so it modifies the A nucleotide, that can flag a transcript to be degraded or stabilized. And so by copying the RNA into DNA, we actually miss a lot of the information, the extra information that might actually lie in those RNA molecules. And it's known potentially based on primarily non-coding RNAs or small RNAs, but we know that in the RNA world, there are over 100 types of RNA modifications. And so when we're, again, doing this copying of RNA to DNA, we're missing out on a lot of extra information about the actual output of the genome. So with this Nanopore sequencing, what you can also do is sequence the RNA directly. So you can take the poly A RNA and then actually sequence out that RNA and also observe potential modifications on that RNA, as well as get full length of the RNA. And so how does this work? So here's another cartoon of the nanopore. And so as the molecule threads through, there's a change in the current signal. And if you have a nucleic acid, this could be DNA or RNA, and it's unmodified, it will give a certain signal. But if you have a modified base, shown here as this red sphere, that will block the current block the pore in a different way, and that will be read off as a different change in current or signal. And so the way this protocol works to actually sequence RNA directly, first you can capture those polyadenylated RNAs. So those are just RNAs that have a long poly A tail at the end. And then you can add this adapter to it that ligates to the very end of that RNA molecule. And then typically, this step is optional, but it seems to help stabilize the molecule and also allow for more throughput in the sequencing. But you basically make an RNA-DNA hybrid. So you get one RNA strand and one DNA strand. And again, that just helps to stabilize or what's thought to help stabilize the molecule. And then at the last step, you can adapt ligate the adapter for the nanopore sequencing, which includes this motor protein that helps feed the molecule through that pore. And because of this library preparation method, you end up pulling the RNA molecule through the pore. So in collaboration with 5 other institutes, so this is a very international consortium, consists of a lab in Canada and a few labs in Europe. We took a particular human cell line. It's a cell line that's used as a reference for many other studies. And we wanted to sequence the RNA directly from the cell line and do a lot of analyses and ask what can we learn from this type of RNA sequencing. And I mentioned bioRxiv, so I'll also highlight again that this work that I'm highlighting is also on the preprint server. And so here's just one data slide I'm going to show from that initial work. So again, we can take this RNA sequencing data, and we can use our FLAIR computational pipeline to then analyze this RNA sequencing data to find isoforms that are expressed in this cell line. And so here I'm showing a diagram of 2 isoforms that were identified from our RNA sequencing. And in red, I'm marking a location that was known to be modified in this way called M6A. So it's methylating the A nucleotide in the RNA. And so one isoform is about 2,000 nucleotides long, and the other one is about 1,600 nucleotides long. And if you notice, the modification occurs over here to the left of the transcript, but the transcripts look exactly the same until you get to the other side of the transcript over here, where it looks like in one case there's an intron that's spliced out, and then the orange or the top transcript, that intron is not spliced out. So the modification exists about 1,000 nucleotides away from the part of the transcripts that distinguish between the two. And so if we were to use some sort of short read technology to detect any differences in modifications of these 2 transcripts, it would be really difficult because the thing that makes them different is on the totally other side of the molecule. So, but with Nanopore direct RNA sequencing, we are able to see a difference in the types of current that's happening at this location. And because we have long reads, we can more confidently assign which RNA molecule had that type of modification. So this graph I'm just showing is a distribution of all the current signals that came out from this position of all the reads that were assigned to the orange transcript, and then all the reads that were assigned to the green transcript. And you can just see that they're a different distribution. And again, the only reason we were able to really tell which read goes to which transcript is because we had these long reads. So that was just some preliminary work. This was done in a human cell line. But a lot of our future work that we're interested in moving forward in the lab is to actually do this native RNA sequencing directly in primary cancer samples. And again, the reason is we already know that cancer cells will alter the transcriptome either through gene expression, splicing. It's known to also affect the way RNAs get modified. But being able to actually use one sequencing platform with the long reads to get all of that information and get the full complete picture of what the output of the cancer cells are will be really powerful in identifying maybe new ways that the RNAs are altered in cancer. And so we're interested in, and this is definitely future work and a lot of work in progress to get to this point, but definitely this is what we're interested in doing in the future. So with that, I have my own acknowledgement slides as well. So again, really like to thank Cameron. He was my first graduate student in the lab. I was very lucky to have such a talented student and also very knowledgeable in bioinformatics and RNA biology. And he's also funded by the Ford Foundation. So we've been fortunate to have a lot of funding from private foundations, as well as NIH training grants for the lab. And also I mentioned work also from Allison Tang, who also helped with the FLAIR development and also the native RNA sequencing collaboration that we had. And then that native RNA sequencing project I mentioned, again, is a large collaboration, a large international collaboration, and it's been really led by Mark Akeson here at UC Santa Cruz, as well as Winston Tipp at Johns Hopkins. So that's all. We have other projects in the lab, but I just wanted to highlight our work with Nanopore sequencing. And if you want to read more about our published work or our bioarchive work, you can check our website. We also have a Twitter handle. One of my graduate students is my social media account manager, so he's way better at Twitter than I would be. So, but we tend to post things that are happening in the lab. So thanks for your attention. We can take questions now.
Currently, what is the accuracy level of the RNA sequencing?
It's about the same. So we get about 10% error rate as well. One error that we are seeing that's a little different than with cDNA sequencing is we're seeing a lot more fragmentation of the RNA, and we don't know if that's the preparation, or we don't think it's happening during the sequencing process, so we think it's something about the way we prepare the RNA. So I know Mark's lab is really working heavily to try to change the protocol to help with that.
Thank you.
Yeah.
Can you speculate on why an error in splicing can lead to cancer?
Yeah. So there are many reasons why an error in splicing can lead to cancer, but specifically for splicing factors, it's very reasonable to think that they're contributing to cancer because they impact so many genes. Like, for example, the U2F1 gene that I'm looking at, it splices every single intron, so it's essentially touching every single gene in the transcriptome. So it's feasible that some of the genes that it's altering are involved in cancer-related pathways. And so for my project, that's kind of what I'm hoping to find.
Yeah. Next. Yeah. Wait for the mic. Oh, there you go.
Thank you. I have two questions. One is about nanopore sequencing. So, like, you said the error rate's about 10%, but how do you establish the error rate? Like, do you have like a quality factor or confidence? And the second question is about the isoforms. So I think, Cameron, you said like you had 1/3 that were like novel isoforms. So I just wanted to wonder how much of that could be like because of an error in sequencing.
Yeah, so to answer your first question, the error rate is established through alignment. So when you sequence out a molecule, you'll align it back to either a reference transcriptome or a reference genome. And so the calculation for how many errors are actually in that read is very simple. It's just the number of matches over mismatches and insertions and deletions.
And it's pretty consistent. Like every nanopore experiment, whether it's DNA or RNA, kind of has the same error rate. So
there are some errors that are systematic in regions like homopolymers. You tend to have a lot more errors in those regions, but Nanopore or ONT is releasing new software to help overcome that systematic error. Sorry, can you repeat your second question?
So for the isoforms, you said 1/3 of them were novel. So like, how much of that could be an impact of the error?
Yeah. So an early step in the FLAIR algorithm, we do a lot of filtering to make sure we don't include, to basically reduce our false positive rate. So there's an early step where we do filtering based on how good an alignment was, which takes into account things like how many bases were misaligned, how many mismatches there were, how many indels there were. So we haven't looked into false positive rates, I don't think, for...
a little bit. So it's more in the bioRxiv paper. So one, we've assessed it through simulated data, as well as there's these spike in RNA molecules of known sequence and abundance. And so we also use that to kind of benchmark the tool. But actually, one common issue that we've found with the error rate is it's really hard to do what's called a spliced alignment. So usually when people do RNA sequencing, they try to align the RNA molecules to the genome. But since the genome contains the intron, but that intron was spliced out in the RNA, there's actually a huge gap in how to match up the RNA molecule to the genome. And so there are other tools to do that computational step. And it's really hard to do when you have a lot of errors. So we were concerned with that. So the way we've dealt with that is to basically rely on what are annotated splice junctions. And also, since the short read data, it's short, and it's very accurate, but it's also good at finding individual splice junctions. So just one, it's kind of easier to map one intron. And so we use that information to be more confident about what splices actually exist in the Nanopore data. Go back and then, oh, is there one? Oh, sorry.
Another basic question. Thank you. Can you explain to us how the pathologist determines the genomic score? And for now, what does that tell the specialist oncologist or radiologist oncologist on what therapies to follow besides the inherent level of aggressiveness in the tumor?
So what do you mean by genomic score? Maybe I don't.
Genomic score on a scale from 0 to 1.
I'm not familiar with that terminology, so I'm not sure. Or is it that you're asking, like how much the genes are mutated? Is that kind of what you mean by genomic score?
I believe that's what it means.
Okay, yeah, I'm actually
not
not
familiar. The higher
familiar.
it is close to 1, the more aggressive it is.
Of the genomic score. So
It takes a month after the regular pathology to come back.
I see. So I'll say the caveat, as I'm not a pathologist, so that's part of why I'm not as familiar with that terminology. But I know, so at least for lung cancer, it's more and more common practice to look for certain genomic characteristics that maybe goes into a genomic score. So for example, that the EGFR mutation that I mentioned, that's fairly standard of care. If someone comes in with lung cancer, they'll get that gene sequenced because not only are there targeted therapies for that gene, but actually it's been shown that if you give those patients standard chemotherapy, they actually do perform worse. So it's actually really important that you know the status of that gene. So I could imagine that might come into a score. There's also, many of you might have seen commercials for like Keytruda, or there's these new immunotherapies that have come up that's been really amazing in terms of treatment and has impacted a lot of patients. And so I know that there are certain biomarkers that are surveyed that, for example, the expression of a certain gene that is actually the gene that's targeted by these drugs. And so those are also, I think, I don't know if that's standard of care yet, but I think there are a lot of clinical trials to actually sample, how much this gene is expressed, and then treat those patients with certain immunotherapy. Yeah. Back there.
Hi. So when you're directly sequencing the mRNA, there are hundreds of different modifications that occur. What are your thoughts on how do you identify what exactly those modifications are? Will this technology allow you to identify them directly, or are you going to have to use some type of orthogonal method once you've identified where a modification occurs?
Yeah, so one challenge, and so the way, to kind of go back of how the technology works is you actually have to train the model, the computational model that takes that current, those squiggles of current that's occurring, and translate that into an actual sequence. And so right now, one of the challenges is if there's a modification there, we don't know what that current should look like. And so right now, our group, many other groups are trying to build training sets, meaning create synthetically generated RNAs with a known modification, and then pull that through the pore so we can map exactly what current signal will match with what sequence. So that's one challenge. The other challenge is the way these algorithms work. Once you expand the alphabet, it'll be very challenging to then really, be able to notice all the different subtle changes and how that maps to different modifications. So, I imagine initially, just like what we were doing with the isoform assembly, we kind of use orthogonal methods like short read data where we're more confident. There's other kind of orthogonal methods you can be more confident about exactly where a modification should be, and that can help kind of train these algorithms and learn more from them. But yeah, it's going to be a big challenge because 100 plus is a lot, but definitely we want to try to get there. Maybe there was. Oh.
So when you see these new RNA isoforms, you could see known splice junctions that are assembled in a new and different way, and you could see entirely new splice junctions. So what are you seeing?
Yeah, I think Allison has done the most comprehensive
Yeah.
analysis of that, but we definitely get a mix of both. So as Angela mentioned, we're using short read data to figure out what are the possible splice sites for a given gene. And what we've seen is that we do find a lot of isoforms that are using known junctions, just in unique combinations. And then we're also seeing a lot of isoforms that use novel junctions that weren't in any type of annotation, but they were in our short read data. And so I'm not sure what the exact percentages of these different types of isoforms, but we definitely are seeing both types.
One really striking number that I do remember off the top of my head is, at least for the native, so that those numbers kind of came from our native RNA sequencing project. And out of all the transcripts we see expressed, 30% of the transcripts are novel, meaning they're not, either novel combinations of splice junctions or, totally novel splice sites included. So it's a pretty large fraction of the expressed transcriptome, at least for that cell line, that we're seeing are not in our databases. And then maybe after.
In your picture of wild type cell lines, you had some that were two wild type and some that were wild type and the S whatever.
Mutant,
Why didn't you have the 2S?
Yeah, that's a really good question. So something I didn't mention is that U2AF1 is essential for cell survival. If you delete both copies, then your cells are, they're dead. And so this mutation, this S34F mutation that we find in different cancers, it's always present as only occurring in one of the copies of U2AF1.
Maybe, yeah.
Hi. One of the slides you had I thought was very interesting, where you showed the immune cells having large numbers of splicing gene mutations. And I'm somewhat familiar with the, immune cells and how they're different with other like solid cancers and stuff. And I thought you might maybe mention a little bit about the why there's a disparity between splice mutations in immune cells versus solid tumors, for example.
That's a great, I think it's still kind of a mystery why they're more prevalent in blood malignancies compared to solid tumors. The same mutation, so one of the first studies I did as a postdoc, I looked at the U2F1 mutation in both lung cancer and in AML, which is acute myeloid leukemia. And you can see the same molecular changes. You could see the same type of introns that were not being recognized, and even similar genes that were being altered. And so those are some of the other genes we're interested in following up on. But it still wasn't quite clear. You could have the same mutation but have very different cancer contexts as to why they're occurring in both. But that's kind of why we're looking at different splicing factor mutations and these mutations in different contexts to try to maybe see what is similar amongst all of them.
Given your current situation with your database and how extensive it is with all the donor sequences, would you say your future work would be most accelerated by expanding that pool of data you currently have, or working more towards enhancing FLAIR and the algorithms behind the calculation?
Great, that's actually a really great question. I would say, I guess some kind of philosophy when building computational tools, and this sort of from my own personal experience and why we sort of have one tool that's worked on different projects. I feel like when you're building a tool, it's good to use it on many different inputs with different different biological questions and different data types, because I think that gives you kind of the most robust tool. So that's why I would say I'm more interested as a lab to perform more sequencing to collect more data, and that I think will help improve the FLAIR algorithm by having different questions that you need to ask with the same tool. Maybe I don't know. Are we good on time? I don't know. Like maybe one more question. Okay.
Thank you. So for the novel transcriptors you identified, how do we make sure that they have distinct functions from the others? Do we have good method to determine that they actually have distinct functions? Thank you.
Yeah, that's the next step's the part that I'm trying to work out right now. But there's very sparse annotations for isoforms, as you may know. So trying to guess the function of an isoform or even a gene sometimes is really hard to do. And so what we're mainly trying to target are isoforms that come from genes in known pathways. And so with at least that much information, we can test to see if we introduce an isoform from a gene in some pathway, we can see if there is any impact on that pathway, if it's being activated or if it's not.
And we have other projects in the lab that tries to think about how do we actually test like all 300 isoforms all at the same time. And it's, it's definitely something we definitely think about in the lab. Yeah. Okay. And so thanks again. I'd like to say that there's coffee. Yes, coffee and tea. So, and then we're here to answer more questions. So thank you again, everyone, for coming and your attention.